Hi, good afternoon, everyone. Thank you for joining us for today's webinar. I'm Cassie, Marketing Supervisor at Jamscript, and I'll be the moderator for the webinar today. So a little background about Jamscript for those who are new to us. Jamscript is the leading life sciences research and application service and product provider in various fields from basic life sciences research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. The topic for today's webinar is Enhancing Strategies for COVID-19 Solutions in Diagnosis, Vaccine Development, and Therapies. Let me quickly introduce our speaker for today, Dr. Leong Hao Wing. Dr. Leong is the field application scientist supporting the protein expression and antibody production services at Jamscript. Dr. Leong graduated from the National University of Singapore in 2011, majoring in pharmacology and neuroscience. She has more than 10 years of research experience in molecular biology techniques and extensive knowledge in protein structure modeling, protein expression, and its purification. This webinar will take around 40 minutes and we'll have about 15 minutes for Q&A session later. You can type in your questions in the chat box and we'll answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll email the answers back to you after this webinar. So now, without further ado, we'll let Dr. Leong to start her presentation right now. Dr. Leong, over to you.
Okay, thank you. Thank you, Casey, for the introduction. Yes, good afternoon, everyone here. Thanks for taking time off from your busy schedule to attend the webinar. And I'm Hao Wing, and I'm the field application scientist for Jamscript. And it is my pleasure here to share the current trend in the industry in how we could actually enhance the strategies for COVID-19 solutions in diagnosis, vaccine development, and its therapies. So for today's presentation, I will first discuss the current status of COVID-19 pandemic and what are some of the current strategies in tackling and controlling COVID-19. How far are we in getting out of the pandemic and what are the possible future remedies required? Last but not least, how we could use science to provide solutions in hopefully eradicating COVID-19. So started around in December 2019, as such the name COVID-19. It has been nearly more than 1.5 years since its first confirmed report from Wuhan in China. And since then, COVID-19 has spread rapidly, affecting billions of people around the world. And as of today, the total number of reported cases report to be more than 209 million. Cases, causing nearly 4.3 million in deaths worldwide. So for the past 1.5 years, scientists all over the world have been working tirelessly on a multinational effort to identify the novel coronavirus. The SARS coronavirus 2, SARS-CoV-2, has been identified as the etiologic agent of this disease. So the spike protein of the SARS-CoV-2, previously named 2019 nCoV, is composed of the S1 and the S2 domain. The S1 domain contains a receptor binding domain that can specifically bind to the angiotensin converting enzyme 2, ACE2, the receptor on the target cells. It is believed that the SARS-CoV-2 spike protein, RBD, has potential value for the diagnosis of the virus. However, currently, the approved treatment of choice is for this disease, for COVID-19, is just a symptomatic kind of treatment treating and curbing the inflammation caused by COVID-19. As such, anti-inflammatory drugs such as baricitinib and tocilizumab typically used as anti-inflammatory drugs for rheumatic arthritis, or steroidal drugs such as the dexamethasone are used. In addition, as there are currently no targeted anti-SARS-CoV-2 drugs available, the approved antiviral drug remdesivir, broad-spectrum antiviral medication, is used in conjunction with the anti-inflammatory dexamethasone. With it start only in the year 2019, and considering it is a public emergency, the Food and Drug Administration, FDA, has also authorized the Emergency Use Authorization, EUA, for some of the unapproved therapies, such as the anti-SARS-CoV-2 monoclonal antibodies like the Casevrimab and the Imdevimab and the Sotrovimab. So in addition, much effort has been embarked on identifying appropriate vaccines to provide protection against COVID-19, with 10 commonly used vaccines of different nature, such as mRNA-based vaccine or adenovirus-based vaccine, or to carry the spike protein, or even peptide vaccines. In addition, there are more traditional vaccines that uses an inactivated SARS-CoV-2 to create the immune response on in the vaccinated person. So despite progresses in providing targeted therapies and vaccines, the SARS-CoV-2 is rapidly mutating and increasing its transmissibility and also increasing the severity of the disease. So currently, 4 major mutant variants have been identified up to date, with the new one coming up, the Lambda also coming up. But I have highlighted here the 4 major ones with the B1.17, the alpha strain, the B1.351, the beta strain, and the P1 gamma strain, and of course the most recent B1.617.2 delta strain. Due to its high virulence, such strains have also spread rapidly, or even more rapidly than in some instances where it first originated, to many other parts of the world. So the aforementioned variants are due to key mutations in the spike protein, such as the N501Y at the RBD of the spike protein. This mutation is found in all 3 of the mutant variants, the alpha, the beta, and the gamma. And in addition, E484K and K417N mutations are seen in the B1.351 and the P1, for instances. For the recent Delta strain, 2 other mutations are also observed. That is the E484Q and the L452R are observed. Such mutations are observed either to increase the ACE2 binding, escape the host immune system, or enhance their ability to replicate, or it could affect the furin cleavage in increasing exposure of the spike protein to the ACE2 binding. As such, there have been concerns that the current vaccines available may not be efficacious against some of the mutations and the strains. And as such, much more studies have to carry out to ensure the efficacy of the therapies and also the vaccines, and also preventive measure against the new COVID-19 variants. And also work has to be done to ensure that we provide solutions to keep up with the ever-changing viral genome. So in Jamscript, as a partner for research scientists to embark in making research easier, we have several platforms and solutions for SARS-CoV-2. We have products and materials that could be used for nucleic acid assay development to make RT-qPCR diagnostic kits quickly to quickly identify the source of COVID-19 and to curb its spread. We have proteins that could be used in serological assay to determine the efficacy of the vaccine in vaccinated individuals. In addition, such proteins can also be used to study the mechanism of the different variants. And also we provide cell lines for drug discoveries against COVID-19. And of course, our extensive COVID-19 reagent antibodies not only could be used as a good antibody pairs in sandwich ELISA diagnostic kits or even lateral flow diagnostic assays. They could also demonstrate a source or point of study to further optimize as a therapeutic target. And last but not least, our extensive COVID peptide pools and libraries ranging from research rate to to clean rate can also boost with the vaccine development and its validation. So as indicated in my previous slides, the understanding the mechanism of action is crucial in identifying potential therapies. So targeting the cell entry could be one means of a drug therapy. So cell entry of coronavirus, as mentioned, depends on the spike protein binding to a cellular receptor and priming, which was actually primed by a cellular protease. And recent studies have demonstrated that the angiotensin converting enzyme 2ACE2 serves as the surface receptor to bind to the S protein in SARS-CoV-2 and to facilitate entry of this coronavirus in the cell. As such, in Jamscript, we have developed a single clone stable cell line with high expression of these key regulators of COVID-19, such as the SARS-CoV-2 spike protein or the human ACE2 as powerful cell line tools for discovery to treat COVID-19. The ACE2 cell line in Jamscript is validated for stable ACE2 expression and S protein receptor binding. And they are easy to use in binding assays or as immunogens. Some of the applications include neutralization assays to screen antibodies or other molecules that can block a pseudovirus or live virus infection, or it can be used in affinity analysis of the antibody or other molecules to the spike protein. It can also be used as a form of in vitro cell models for investigating SARS-CoV-2 infection mechanism. So in this case study that I have shared here, Rogers and all had published in Science in 2020, had used such ACE2 expressing cell lines to actually evaluate the monoclonal antibodies and their capabilities for neutralizing SARS-CoV-2 pseudovirus. So antibodies are first grouped according to the epitope specificities, and neutralization IC50s are reported in micrograms per liter. Monoclonal antibodies are evaluated for the neutralization of the SARS-CoV-2 pseudovirus using the HeLa ACE2 target cells. So the most potent neutralizing antibodies were those that are actually directed at the RBD A epitope. To evaluate whether the RBD-A epitope that send the ACE2 binding site, they next perform the cell surface computation assay. Monoclonals were first mixed with the S or the RBD protein and measure the binding ability to HeLa ACE2 target cells as a measure of competition to the cell surface ACE2 receptor. So in addition to the ACE2 cell line, we also have the spike protein cell line that express the full-length spike protein that includes the S1 and the S2 domain. So they are also validated for a stable SARS-CoV-2 spike protein expression and human ACE2 binding. And they are available either in the CHO-K1 or the HEC system. So Tan and all in 2020 had published used such spike protein cell line to generate pseudovirus-based virus neutralization test, PVNT. So just a brief introduction on the neutralization tests that are currently used and available. So we have the conventional virus neutralization test, the PVNT, which requires the use of live SARS-CoV-2 to test the efficacy of neutralizing antibodies in preventing the live SARS-CoV-2 in entering the ACE2 receptor expressing cells. Such CVNT actually requires BSL-3 containment facility, and the process of testing is tedious, requiring 2 to 4 days. PVNT, on the other hand, as the name suggests, use a pseudovirus expressing spike protein as the envelope protein to determine the neutralization capability of the antibodies. Such PVNT requires less stringent requirements, so BSL-2 will be sufficient. However, there is still the need to use a live virus and cells. So Tan and all in this study had generated such PVNT using the spike protein cell line to generate the envelope protein and package it as a pseudovirus using delta VSV-G envelope plasmid. And to compare and develop a surrogate, this is to compare and develop a surrogate of VNT in that such tests do not require the live virus or cells, and is based solely on the virus-host interaction of the RBD and the ACE2 receptor on the ELISA plate. So comparing the three types of tests, they are all comparable, of course, while of course the SVNT will decrease the need of a high stringency of a BSL setup, while a PVNT mimic a proper study of a viral entry into the cells and to determine the efficacy of the neutralizing antibodies. So as such, depending on the needs of the research question, so different VNT could be suitable. And of course, as mentioned, to develop SVNT proteins such as the RBD, which have been identified as a crucial interactor for ACE2 binding, will be required. In fact, this study by Tan and al., which was a collaboration with Jamscript, and we have since then developed many of these COVID-19 proteins used in these studies for SVNT use. And considering this, Jamscript has also moved forward and provide various mutant proteins from various variant strains, such as the N501Y in the alpha strain, the E484, and the K417N, and the N501Y in the beta and gamma strain, and also the E484Q and the L452R and the T478K in the delta strain, and which are good for developing, for example, as mentioned before, the SVNT to neutralization antibody drug discoveries. Such proteins are already tagged with HRP for SVNT development. So these mutants are also available without the HRP to allow mechanism of studies. Mechanism of action studies. As mentioned, these mutants work on several mechanisms. For example, to increase the ACE2 binding affinity to the ACE2, or it can help to escape a virus host immune response, or it can increase its replication ability, or they may be the mutants may have advantage in modulating the cleavage by fluorine in order to prime the S protein for the TMPRSS2 processing to review fusion peptide at the S protein for viral host membrane fusion. So the N501Y is one of the key contact residues with the receptor binding domain and has been identified as increasing the binding affinity to the human and the murine AC2. The spike protein mutation 69 to 70 may also help the virus to escape the host immune response. The spike protein mutation D641G also become dominant during also become dominant, and the D641G mutation enhance its replication ability in the upper respiratory tract, transmission ability, and also viral loads in a patient's lung epithelial cells. It may also affect vaccine efficacy and antibody therapy. So there are many of these different kinds of mutants that are available and Jamscript for clients or for researchers to look into the mechanism of action. So in particular, L542R was of particular concern because the B1.429 is possibly more transmissible. And further study has actually confirmed this. And CDC has actually listed the B1.429 and the related 1.427 as variants of concern, and sites saying that they would exhibit at least 20% increase in the viral transmissibility. So we have such wide range of COVID-19 mutants available because we at Jamscript has more than 15 years of experience in protein synthesis, protein expression services, generating more than 10,000 proteins such as transmembrane proteins, kinases, co-expression proteins, cytokines, growth factors, viral envelope proteins, nuclear and hormone receptors, and also antibodies. So we have delivered more than 5,000 recombinant antibody projects with more than 95% success rate. And how do we do that? Because it is through our use of proprietary technologies that we have developed, such as the GenSmart codon optimization technology and the high-density expression platform. This allows us to deliver high-quality products, but also in a cost-effective manner. In Jamscript, we have 4 different expression systems to allow us to have to express good quality proteins at a competitive pricing that can suit your downstream application and also cater to certain needs and requirements of your protein. For example, while E. coli is a cost-effective way to get the protein at cheap costing, it may not have, post-translational modification that could be important for function or binding capabilities of proteins. So we also have the insect and the mammalian system may be a better choice in such instances, but of course this may come with a higher price for the proteins. So that's why in Jamscript we have various choices of COVID-19 proteins of different domains with different tags by different expression system to suit the budget and also the applications of the researchers. And of course, as mentioned earlier on, antibodies for targeting SARS-CoV-2 not only allow the development of serological assays to determine the efficacy of vaccines, but also to determine or track individuals on the protection against COVID-19 after vaccination. Anti-SARS-CoV-2 monoclonal antibodies are a good form of therapy for COVID-19. And considering the variants now widely in the world, transmitting in the world, more antibody discovery against COVID-19 is required. And to enhance the possibility of a success of costly projects such as monoclonal antibody production, and also to give the best value of monoclonal antibodies, Jamscript has several methods. We have time and tested hybridoma technology. We also use innovative technologies such as the B-cell cloning and also the single B-cell sorting to accelerate B-cell selection and also to capture a large B-cell repertoire. So to elaborate on the hybridoma technology that we use, so upon harnessing the animal's immune system to generate antibodies of high specificity and affinity due to their intrinsic affinity maturation abilities, we use reliable and time-tested hybridoma technology to immortalize antibody-producing B cells producing the hybridoma cell lines to generate high-quality monoclonal antibodies. And just by growing them, the hybridoma cell lines would be able to provide us with an unlimited supply of antibodies. And in addition, Jamscript, we also tap on the Jamscript monoRep technology, which enable us to generate a large number of high quality of rapid monoclonals to maximize the success of identifying monoclonal antibodies for research applications such as IHC or in therapeutics or even in diagnostic kits. So due to the rabbit's superior immune system, they can generate antibodies of high affinity, specificity, sensitivity, and stability than those from other animal species. In fact, rabbits they have the robust and unique immune system also enable superior diversity of antibody generation that is up to 10 to 100 times higher affinity compared to the rodents. So just a little bit more about, how the monoRep what's the workflow of the monoRep in Jamscript. So it involves in the isolation and the enrichment and the culturing of antigen-positive B cells directly from rabbit peripheral blood mononuclear cells, PBMCs, for screening and selection. And with our optimized and proprietary technology, we are able to enrich and expand the B cells and use them directly for screening. And as such, it is fusion-free from lymphoma, and it can avoid the B cell loss, and as such, we have a better ability to obtain a large cell pool of up to 10,000 memory B cells. So the desired monoclonal clones are sequenced and recombinantly expressed. And many benefits of this approach include PBMC, such that the animals are not sacrificed. So we can go back to the animal after testing in the polyclonal anti-serum, for example. It is fusion-free, so it will avoid B cell loss and allow us to obtain a larger cell pool. So the lack of fusion also increased the efficiency of discovery, and through B cell cloning, we are able to achieve antibody in a high throughput manner in as fast as 13 weeks. However, despite various advanced technologies, so we also for this one wait, I think I skipped one. Okay, yeah, sorry. So we also have another system. So despite the advanced technology that we have, such as the hybridoma and also the B cell cloning, researchers also require more clones to prevent missed opportunities and even higher success rates, and they sometimes require more time-efficient antibody discovery. Tapping on the recent engineering feat, the Beacon platform, which actually used the optofluidic system to allow the screening of individual B cells in a high throughput manner. Carrying the screening on a chip with different chip assay available, such as the antigen and specific bit assay, or the IgG capture assay, or even the GFP reporter cell assay, it allows improve the speed and also increase the diversity of the antibodies that can be discovered during this antibody discovery step. So in Jamscript, we have also tried to compare, the three different technologies, namely the hybridoma, the Beacon, and the mono-red B cell cloning technology. And we have Jamscript have also joined hands with researchers and have worked tirelessly to generate, high affinity and high diversity antibodies against the antigens such as the RBD binding domain, as shown here. So as mentioned, the spike protein has been recognized as a popular vaccine target to develop COVID-19 vaccine. So many therapeutics are actually leveraging on the finding a neutralizing antibody that can block the RBD and the ACE2 interaction, and thus to reduce the virus infectivity. And as such, high throughput and efficient antibody discovery is needed. So in Jamscript, how do we go about generating, the different antibodies that we have in Jamscript? So first, we actually, after the rabbit immunization, we actually generated a cell suspension from round rabbit spleen, and then subsequently concentrated the antibody secreting cells using the antigen and IgG positive B cell enrichment process that we have. Altogether, we imported about over 15,000 cells onto the Beacon system into each well, as shown here, and subjected to the cells to the antigen positive screening using the antigen beads conjugated to RBD. So eventually, we were able to identify a total of 142 RBD positive hits. So as you can see here, a total of 8 monoclonal antibodies were obtained in the Beacon experiments compared to 3 in the hybridoma approach. And these 8 monoclonal antibodies from the Beacon platform also have a higher diversity, and it span across 4 epitope beans, while all of the hybridoma antibody span across only 1 epitope bean. So the high antibody diversity could successfully enable us to have multiple generate antibodies for multiple different downstream applications, making them excellent candidates for sandwich pairs in vitro diagnostic kits or other assay development. Or they could also be allow researchers as a start for potential development into therapeutics thanks to the many candidates that have the neutralizing efficacy. So here we show our B-cell cloning approach. So for our B-cell cloning, we observe that we are able to also obtain a high number of positive monoclonal antibody clones. A total of 16 monoclonal antibodies were obtained through this B-cell cloning approach, even though we did not load it into the high-throughput system on the beacon. We were also able to have a high diversity that span across 11 epitope bins. So the high antibody diversity could enable us to do many downstream applications. So such antibodies are rigorously tested for their ACE2 binding capabilities and then pseudovirus neutralization capabilities. And so they have already been, as mentioned, to identify the epitope that it is binding to the RBD, and thus it allows us to provide with the best antibody pairing for example, in sandwich ELISA IVD setup. And so they are also have already been tested for Western blot and FACS. So how do we obtain, such antibodies with such different high specificity and affinity? Because of a series of proprietary antibody technologies that we have to ensure the success. For example, we have the optimum antigen design program, which is an advanced antigen design algorithm to optimize the likelihood of surface exposure and authenticity. We also have the Immunoplus technology to make use of key immunomodulators to circumvent self-tolerance mechanisms and to allow B cells to generate high affinity surrogate antibodies. So we also have epitope binning, which is an independent antibody validation in which the monoclonal antibodies are tested against one another to determine which antibodies compete for the same antigen epitope. So Jamscript has also other proprietary adjuvant system to quickly elicit a strong and durable immune response to generate high specific antibodies. And last but not least, we have the express immunization and high efficiency electrofusion to allow antibody generation with a speedy turnaround and at a large quantity and high diversity. So as such, we have available not only towards the RBD, we also have antibodies that target the S1 neutralizing antibodies and antibody pairs of different antibody formats, host for different kinds of applications. And not only so, we also have developed antibodies for the S1. We also have developed antibodies against the non-RBD binding antibodies, binding to the non-RBD region of S1 and S2. So in addition, antibodies binding to the nucleocapsid of the SARS-CoV-2 is also available for different applications. And these are also good as control antibody for COVID-19 studies. We also have neutralizing antibody standard for you to calibrate while setting up your SVNT or PVNT. So as mentioned, with our capabilities earlier on, we have capabilities in peptide synthesis, having one of the world's largest automated synthesizer system, high throughput PEP-H2S library platform that we have in Jamscript. And we also have developed our own proprietary technology, such as the high-syn technology and new pre-algorithm to detect difficult to synthesize peptides and to provide with the technology to synthesize difficult peptides. So we are well equipped to synthesize quality peptides for COVID-19 research. So not only so, we are ISO 9001 certified, and we have quality management providing MS and HPLC analysis as QC for our clients. And in addition, we provide a solubility testing, also a TFA counter ion removal, endotoxin control for our peptides. So how could peptide be important or could help us in our COVID-19 therapeutic development? So in this study by Ma and al., they have investigated the production of nanoparticle vaccines by covalently conjugating the self-assembled 24-mer ferritin to the receptor binding domain RBD. And or the heptide repeats subunits of the severe acute of the SARS-CoV-2 spike protein. Next, using the many different peptide pools targeting the spike glycoprotein, Ma and all have validated that the nanoparticle vaccines elicited more antigen-specific polyclonal, polyfunctional CD8 plus T cells expressing the IFN gamma and IL-2 and the TNF alpha than the monomer vaccines in the intracellular cytokine staining ICCS assays. In addition, a previous study on SARS-CoV-2 has also revealed that the type 1 T cell T helper cell Th1 bias immune response enhanced protection against the virus infection, while the Th2 bias immune response induced vaccine-associated enhanced respiratory disease. So they measured the percentage of IFN gamma and CD4 plus T cells, the TH, which are the Th1 bias cells, and the IL-4 CD4 plus T cell, which are the Th2 bias cells, in using ICCS across the different vaccine group. So they found that the nanoparticle vaccines induce a higher Th1 based IFN gamma compared to the monomer vaccines. And the percentage of Th2 bias were also no difference across these groups. So T cell responses were further confirmed by ELISA spot assay, indicating that the RBD and the RBD-HR nanoparticle vaccines are able to induce strong and safe T cell immune response apart from B cell responses. So peptide pools generated can be used for vaccine development and their validation in, for example, as shown here, ICC assays like this. So as mentioned, these are the different peptide pools available targeting some of the crucial gene, spike protein and spike protein harboring the mutants for your research. In addition, we also have peptide libraries for various spike protein created using overlapping libraries to allow researchers to map epitope of interest and also to stimulate a T cell in T cell assay, or to also either map the binding sites of spike protein with ACE2 of a different purity depending on the applications. We have various collaborations with researchers and also have many expertise of peptides and the applications. Thus, we understand the challenges while working with a T cell activation assay. That is, for example, the requirements of high purity to ensure no specific activation of T cells, or the need to have a low level of TFA to ensure low toxicity contributed by these chemicals during a T cell assay. As such, we have assurance for guarantee TFA removal and also high purity peptides with stringent QC. As mentioned, we also have single peptides available that could be good for generating a peptide vaccine or for eliciting antibody responses in animals for antibody discovery against COVID-19. Last but not least, besides the various ways we could help researchers with COVID-19 research, vaccine validation and development, diagnostic kits are also crucial to ensure the quick identification of COVID-19 clusters to curb their spread. So several detection methods are available, such as the nucleic acid detection or the serology detection using antibodies. So in Jamscript, in addition to the PVNT, SVNT, and also providing antibodies for IVD kits development, we do have the COVID-19 molecular diagnosis platform available. So using the 4 target genes that are highly specific for the SARS-CoV-2, namely the ORF1AB, the RDRP, and the ENN gene. We have developed oligonucleotide primers and probes against these genes for use in RT-PCR assay, in which the RT-PCR is recognized as the gold standard for COVID-19 detection. So a typical workflow will include the following. That is the sample collection and the viral extraction, followed by the QRT-PCR, followed by results validation. So these are the primers that are available in Jamscript. So the different standard that could fulfill your need, that is for research use and also for clinical use. So with all the information put together, I hope I have shared some of the ways in which we could enhance the strategies in developing COVID-19 solutions for accurate diagnosis and vaccines and therapies using the technologies that we have here in Jamscript. Thank you for your time. Yeah. Any questions?
Thanks, Dr. Leong. Yeah, thanks, Dr. Leong, for the great presentation. We have some questions from the floor. Let's just answer them. But for other questions, you can still put in the questions on the questions box. So the first question, can you synthesize peptides with all D amino acids? If yes, can you provide me a rough idea of turnaround time?
Okay, we do have synthesizing peptides with D amino acids. So do send us your sequence, because it will depend on the purity and also the purity and also the total length of the sequence. So with a longer length, of course, there will be a longer time that is required, purity also, and also we need to understand, the hydrophobicity of the peptide. So do send us your sequence. Incorporating D amino acids is available for peptide synthesis in Jamscript.
Okay, thanks a lot, Dr. Leong. For diagnostic and detection of COVID-19, are these kits received in CVIVD or FCS already?
Can you repeat the question?
Oh, yeah, sure. For diagnostic and detection of COVID-19, this kit was received CVIVD or FCS already?
So you are meaning the kit that are available? We provide the tools for building up the kits, if I didn't understand wrongly. So we have, like, for example, the primers that are available for building up the kits, the RT-PCR diagnostic kits for COVID-19. We also have the antibodies that are available to build up the ELISA or LFA flow kit that could be used for your this kits generation.
Okay, thanks, Dr. Leong.
Yeah.
I hope we done. I hope we did. Okay, for the next question, what is the purity of the protein antigen that's acceptable to you? What buffer should I use?
In what kind of application? Because it will differ the purity that is required for antigen. That is, I assume this question is for antibody generation. Is it for antibody generation?
Yeah.
Yes, okay, for antibody generation. So what is the purity, is it?
Yes, correct.
At least 85% purity. Usually
okay. What is the buffer that
will be best? PBS. PBS.
Okay, thanks a lot.
Buffer. That will be, that is the usual one that we use.
Okay, thanks a lot. The next question, what are the likely success rate for the use of spike protein peptides for passive immunization?
What is the, sorry?
What are the likely success rate for the use of spike protein peptides for passive immunization?
Passive immunization. I think it will depend on the final downstream application in terms of what is the readout that you are interested in looking at. So yeah.
Okay, maybe we can answer that in more details after the presentation. Okay. The next question, what kind of antigen that you can accept for hybridoma generation?
What kind of antigen that I can accept for hybridoma all kinds of antigen?
What kind of antigen that you can accept?
Yeah, it can be any kind of antigen. It can be peptide, it can be a protein, it can be heptin also.
Okay, can.
Yeah.
The next question, what's the difference? Do you still have anything else to add on for the previous question?
Yeah, it can be, I don't know, if protein can be human, it can be mouse and rabbits, you know, depending on like for example the host like for example the percentage homology with the mouse. So this one we will evaluate, you know, to advise on the best system to use, like whether it's the mouse or the rabbits, depending on the homology. Yeah, so yeah, we can do all kinds of antigens.
Okay, thanks a lot, Dr. Leong. The next question, does Jamscript provide virus-like particles for SARS-CoV-2? If yes, may I know the overview of the construction process through email?
Currently, we do not provide virus-like particles. Yeah.
Okay.
Not at the moment. Yeah.
Okay, thanks. I think let's answer the last question for now. What is the difference between protein AG purification and affinity purification in terms of yield?
Of course, protein AG, right? It recognizes the FC region. So theoretically speaking, you know, when we immunize the animal, you know, the majority of the antibodies that I produce that is going to be purified is against the antigen of interest. But of course, there will still be, you know, some that may be, you know, not specific to the antigen. So of course we have affinity purified ones which is specifically to the antigen. So of course the yield in terms of that will be slightly lower compared to the protein AG. But if it is carried out correctly, if the background is not too, that means your antigen compared to the background of the animal is not too high, then of course it should be comparable. So it really depends on the target of interest of the antigen, the background of the animal. That, like for example, if you want it to be a mouse antigen with a mouse, then of course, you know, there may be some issue, you know. Yeah, so yeah.
Okay, thanks a lot, Dr. Leong. So if there's no other questions from the floor, we'll close the session today. So feel free to email us if you have any other questions after this session. So thank you once again, Dr. Leong, for the presentation, and to all of you who joined us today. I hope you'll find this session beneficial to your work. So thank you, everyone, and have a nice day.
Thank you. Thank you. Thank you for your time.
Thank you. Bye-bye.
Bye-bye.